Hello, my name is Julian Gilmore. I'm a graduate student in the lab of Dr. Raina Bendayan at the University of Toronto Leslie Dan Faculty of Pharmacy. And today I will be sharing with you the work that we've been doing looking at the interaction between dolutegravir and folate transporters and the folate receptor in human and rodent placenta. Dolutegravir is a drug of the HIV Integrase Strand transfer inhibitor class, which is widely used and is rapidly becoming a preferred component of many first-line antiretroviral therapy regimens due to its general safety, its low cost, and its tolerability, as well as to its high barrier to resistance. However, recent studies, including the Tsopamo study out of Botswana, have identified a risk of neural tube defects in the pregnancies of women who are taking dolutegravir from the time of conception. In the results from the Tsopamo study, we can see that the rate of neural tube defects was 0.94%, which was considerably greater than the rate of neural tube defects in the births of women from the same study who were on antiretroviral therapy regimens without dolutegravir. This rate of neural tube defects was reduced to 0.3% in a follow-up study, but was still 3 times higher than what was seen in pregnancies of women who were on other antiretroviral drugs. Since then, other studies have followed up on these findings, indicating perhaps a weaker association between dolutegravir exposure and neural tube defects. Importantly, in Botswana, where the study took place, folate supplementation of grains is not mandated. Due to the established association between neural tube defects and folate dysregulation, these findings raise questions as to whether dolutegravir could be interacting with or impairing folate delivery. Folates, a family of vitamins obtained in the diet through leafy green vegetables and fortified grains, are essential in pregnancy, especially during neurogenesis in the first trimester. Potential folate dysregulation associated with dolutegravir has been investigated somewhat at the clinical level, but information from a mechanistic perspective is still limited. One study from 2019 did identify that dolutegravir could inhibit binding of folates to the folate receptor. However, another study, also from 2019, identified that dolutegravir does not interfere with folate transport at therapeutic concentrations. The findings of these studies and others led us to investigate how in utero exposure to dolutegravir could be impacting mechanisms of folate delivery, not necessarily through direct inhibition of folate transport, but potentially through altered regulation of folate delivery mechanisms. There are 3 mechanisms of folate delivery and folate transport across the functional barrier of the placenta, the syncytial trophoblast. The primary mechanism of folate uptake is through the folate receptor, particularly folate receptor alpha or FR-alpha. Expression of the proton-coupled folate transporter PCFT, as well as the bidirectional reduced folate carrier RFC, also contributes to folate transport at the placenta at both the maternal and fetal-facing membranes of the syncytial trophoblast. What we aimed to accomplish in this study was to characterize the expression of these 3 mechanisms of folate transport following exposure to dolutegravir using 3 different models of the placenta: immortalized placental cell lines, human placental explants, and an antiretroviral drug-exposed mouse model of pregnancy. These models together allow for a holistic view of potential interactions between dolutegravir and folate uptake at the placenta. We first investigated the impact of dolutegravir exposure on the expression of folate transporters and the folate receptor in both the choriocarcinoma cell line JAR and in the immortalized first trimester placental cell line HTR8-SV-NEO at clinically relevant concentrations around 500, 2000, and 4000 nanograms per milliliter. We identified a modest but significant downregulation in the expression of the target genes, which I'm not able to show in much detail here, but which led us to investigate any potential impairment of transporter function that could be associated with this downregulation. So on this slide, I'm sharing with you the data from our in vitro transport assay. To look at the function of the reduced folate carrier RFC, we treated the human choriocarcinoma cell line JAR with dolutegravir and identified that 3-hour dolutegravir exposure was in fact associated with impaired uptake of the RFC substrate methotrexate. We repeated this assay looking at the function of the proton-coupled folate transporter PCFT again in the JAR cell line, but didn't identify as much of a significant impact of dolutegravir exposure on uptake of the PCFT substrate folic acid, except at the higher concentration of dolutegravir following the 6-hour treatment. And finally, we want to look at the function of the main uptake pathway of folate in the placenta, that being the folate receptor alpha, and we did in fact identify that dolutegravir treatment impairs the uptake of the FR-alpha substrate folic acid in the JAR cell line both at the 500 and 4000 nanogram per milliliter dosage of dolutegravir. and this was a significant but modest apparent impairment of FR alpha function. We next chose to look at the impact of dolutegravir exposure on the expression of folate transporters and the Photoreceptor in human placental explants derived from healthy donors, again at both 2,000 and 4,000 nanogram per milliliter doses of dolutegravir. What we found following treatments with the higher dose of dolutegravir was a modest upregulation of RFC at the mRNA level, accompanied by a modest downregulation of the Photoreceptor alpha in human placental explants. None of these significant differences were observed at the lower concentration of dolutegravir. The third and final model that we looked at in this study was a pregnancy mouse model of antiretroviral therapy. In this model, we treated mice once daily with doses of dolutegravir that achieved clinically relevant plasma concentrations in the dams as previously characterized in the lab of our collaborator, Dr. Leonard Sergides, again at the therapeutic dose, or a high dose, in this case 5 times the therapeutic dose. We treated these animals either for 10.5 days of pregnancy, right around the time of neural tube closure, or gestational day 15.5, which represents a later stage of neural development. In addition to our in vivo model reflecting chronic dolutegravir exposure, which is experienced in the context of antiretroviral therapy during pregnancy, we also wanted to be able to mirror conditions that are experienced in parts of the world where folate supplementation in the diet during pregnancy is not common, including in Botswana, where the SEPAMO study initially identified this association between neural tube defects and dolutegravir from conception. And so in order to do this, we repeated the study both with animals on a normal diet, but also with animals on a folate deficient diet. What we see here are findings from the animal study, particularly looking at the folate deficient condition, because in this condition we identified some striking impacts of dolutegravir exposure on the expression of the folate transporters and receptor. What I want to highlight in these data is that both at gestational day 10.5 in the top row and gestational day 15.5 in the bottom row, we identified a significant upregulation in the expression of both of the folate transporters RFC and PCFT at the higher 4,000 nanogram per milliliter concentration of dolutegravir. Importantly, at the early time point, gestational day 10.5, where the fetus is most susceptible to issues surrounding neural tube closure, we identified a significant downregulation in the expression of the folate receptor alpha, again at the higher concentration of dolutegravir. And before moving on, I want to mention that we repeated this study in animals on a folate sufficient diet, and the findings that in that condition were not as significant or striking as what we identified amongst animals on the folate deficient diet. So to summarize what I've been discussing, in each of the 3 models I shared with you, we did identify a modest but significant interaction between dolutegravir and the folate transporters and folate receptor expression in the placenta. However, some of the findings we identified vary between the models. One of the highlights in the data that I want to focus on was this downregulation of FR alpha in the folate deficient condition of the mouse model, which is significant for one, because we found this both in our in vivo mouse model as well as in our ex vivo treated human placental explants, and two, because FR alpha is the primary mode of folate uptake at the placenta and plays an essential role in folate delivery to the embryo. I also want to highlight this altered regulation of the folate transporters RFC and PCFT, the directionality of which did vary somewhat between models, but was something that we did consistently see in the cell lines, the explants, as well as in the animal model. Notably, these findings of altered regulation for all 3 genes occurred primarily at higher concentrations of dolutegravir, still within the therapeutic range, but perhaps suggesting that the exposure concentration may be of significance. Finally, I want to highlight this finding of impaired activity of the folate transporter RFC and the folate receptor FR alpha following dolutegravir exposure in our in vitro transport model, which really suggests that dolutegravir could be having an impact on the ability of the fetus to uptake folate across the placenta. And it will be really important to verify this finding in vivo by looking at folate concentrations in the fetus. Ultimately, a lot more work is required to better understand and follow up on clinical findings regarding the use of integrase inhibitors during pregnancy and potential interactions of these drugs with folate delivery mechanisms at the placenta. Before I close, I'd like to thank my supervisor, Dr. Reina Bendayan, and our collaborator, Dr. Leonard Sergides, as well as members of the Bendayan Lab and the Sergides Lab who contributed to this work. I'd also like to thank the University of Toronto Leslie Dan Faculty of Pharmacy and the University Health Network, as well as our funding sources at the OHTN and CIHR. And thank you for your time, and I welcome any questions.